Hello. Since we can't be in person the first week, I'm going to record the introductory microbiology labs. Normally, I'd see you guys on the very first day, but this is the way it is for right now. I'm going to go ahead and record this. So my name is Joshua Horton. I am the microbiology lab coordinator for 4102 and 1102 labs. You guys will see me on occasion coming in and out of the lab, but for the most part, you'll see your TAs. We're going to go over kind of the introductory information. So I will hide my face now so you don't have to look at it forever. And we'll get started with the rest of the lecture. Okay, so this first lecture covers kind of the basic introductory and safety stuff. So, like I said before, my name is Joshua Horton. I'm the lab coordinator. Depending on which labs you're in, you'll have different TAs. For 4102, you guys will all have Shinyi. She will teach sections A, B, and C. She'll be your primary point of contact for a lot of questions, anything you have like that, you'll go to her first. If you're in 1102 in section A and C, you'll have Andrew. And if you are in 1102 section B, D, and E, you will have Liz. The TAs will post their office hours and emails so that you guys can contact them. You can also contact them through Canvas. They're listed as the TAs. What we're going to be covering today. So we've got a lot of things to go over. The primary one, which is actually pretty important, is the syllabus. So we're going to just briefly go over the syllabus. It is available on Canvas, so you guys can download it and look through it on your own. But we will be covering just kind of important parts of it. We'll also be going over safety training, so the initial safety training stuff that we have, as well as the CDC Salmonella case study. Now, really quickly, let's go over that syllabus stuff. You can find it on Canvas. We will probably make some changes throughout the semester. Sometimes dates get marked a little wrong, or sometimes we have to change the labs. So keep that in mind. The calendar of labs and when you will need to attend labs and which items are due are available on that. So make sure you guys bring it with you every single day. It's very, very helpful. There is some listed requirement equipment on there. You do not need it until week 4 and 5. So weeks 1 and week 2 and 3, we won't need that lab equipment, but we will assign you guys drawers and cubbies on week 4 and 5. And what we'll do real quick is we're going to look at the syllabus. Second. All right, so here is the syllabus. You can see it's got lots and lots of information on it. Couple key things. We do need 2 lab books. There's the Microbiology Lab Procedures, which has all the information that we do in the lab, as well as the Photographic Atlas. The Photographic Atlas you don't have to bring with you every single day, but you do need to have it. And it's a very useful resource, especially when you're trying to figure out what kinds of things you need to do and look at. So make sure you guys have that with you. It also tells you kind of like examples of all the stuff and all the tests that we do in the lab. It's a very, very useful resource, not just for the lab, but also for the class that Dr. Lawson teaches. Lab supplies. Important one is a lab coat. It should be 3/4 length sleeve with or cuff sleeves. It basically should go right about to where your wrists are. You don't want to have a very long lab coat that goes over your hands because obviously it gets in the way, but you also don't want it too short. A lot of people will sometimes get things like pharmacy or nursing lab coats, which end at about the elbow, which isn't enough coverage. So make sure you get one that goes all the way down to your wrists. Also get one that goes about midway down your thigh, so it gives you enough coverage from any splashes or anything like that. Lab coats can be purchased from the bookstore or uniform place here in town or lots of other places online, what have you. The lab coat, though, is something that has to stay in the lab. We consider it potentially contaminated. So if you need a lab coat for, say, another lab, let's say you're in chemistry or something like that, what you'll do is on the first day that the lab coat's due, you'll bring your lab coat in and show us, hey, look, I've got a lab coat. It's here, but I have another class that requires a lab coat. I'm not going to make you buy 2. We have some loaner lab coats that you guys can have, and you can just keep them in the lab. And that's where they'll stay until the end of the semester, when we will autoclave them and wash them for you, and you guys can pick them up at the end of the semester. You also need to bring in something that's safety goggles. Now, you want to get the kind that have little vents on the top and bottom, not the kind with holes that are drilled in on the side. So you want to avoid things like impact glasses or the goggles that have little tiny holes drilled on the side that are more like for chemistry or other things like that. You want good splash protection for this one. You can pick these up at any kind of place. It should be, you know, you can get them at the Baylor bookstore, you can get them at hardware stores, you can get them at medical supply shops, you can get them on Amazon, things like that. So there's plenty of places. Safety goggles are another thing that we consider potentially contaminated. So same kind of deal. If you bring them in and you need them for another class, I'm not going to make you buy a second pair. We have some spare ones, so just let us know, but they should stay in your drawer at all times. You should also be wearing nitrile gloves. Nitrile gloves, we generally don't do latex gloves in case anybody has an allergy nearby you or what have you. We do nitrile gloves. You can get them that fit your hand size the best, small, medium, large, or extra large. The one size fits all doesn't work very well in lab, so you want to get a specific hand size. You also don't want to get the ones that are like the clear plastic gloves that are the polyvinyl food handler gloves. Those break way too easily and don't provide enough coverage. One box of gloves will probably do you for the entire semester, so make sure you bring them in. Don't share them with your lab partner. If you like split them with your lab partner and your lab partner is not here, it's locked in their drawer, then you're kind of out of luck and they're not following safety procedures. So make sure you guys have that on. Last little bit is face mask. Obviously, should be worn properly and at all times. So make sure it's over your nose, not hanging off. Make sure you're also not touching it with your hands or your gloves, which is, of course, really bad idea in this class. So make sure you guys keep that in mind. The lab section lists are down here. These will be meeting on alternating weeks, depending on which lab group you're in. And we will have that posted in Canvas so that you guys can look up which lab group you're in, and you guys will know. So the very first week will be those who are in the red, blue, and white groups. And the second week, the B week, will be people in the orange, green, and yellow groups. Lab lecture will be online only. The lecture videos will be posted on Fridays, and you should be able to watch them that day. On top of that, sometimes we'll put up extra resource videos as well, so examples of how to do things in lab, which come in very handy. I do have office hours, which are Fridays, 11 to 12 p.m. in the BSB. A231 is my office. If you want to come and meet with me, shoot me an email and set up an appointment time. Especially if this time doesn't work for you, I have lots of open and available hours. Just shoot me an email. I just have to make a set of office hours. But don't just show up to my office and expect me to be here because sometimes I'm off doing other things like making media or in different parts of the building. So if you do want to meet, shoot me an email. If you want to meet through Zoom, that's totally fine as well. I use Zoom all the time, so just shoot me an email and we can set up a Zoom time as well for that. All right, grading stuff. So exams are about 50% of your grade. There's only two exams. There's a midterm and a final. So that's a very large chunk of your overall grade. So make sure you guys are very careful with that. Similarly, we have quizzes, which are a very high percentage of grade. These are both going to be done only through Canvas. So you'll take the quizzes, and the quizzes are generally going to be open on Fridays for a short window of time. The midterm and the final are going to be open on Fridays from about 8 a.m. until midnight. So there's a big window available in case you guys need to take them at different times. But keep that in mind. There will be no makeup quizzes and exams unless you are, for example, in a university excused absence, so you're traveling somewhere and you can't actually access the Canvas page. Keep that in mind. That's a very rare situation, but it does happen from time to time. Single and mixed unknowns are 15%. We'll talk more about those later, but basically you guys will be doing an unknown and identifying it. We'll talk more about how to do that in a bit. So you guys don't have to worry about right now. Just know that it's a good chunk of your grade and to make sure you follow it. Bacteria Lab is 5%. It's a bacterial identification program that we have. You download it and you guys can operate it on your own on computer. It's like a Canvas quiz, except instead of it being like a quiz per se, it's more like a essay question that lists which organisms you're going to be doing, and then you have to identify them. So that's how that one's set up. We also have lab reports, which are a large chunk of your grade. This includes both in-lab lab reports, which we will hand you guys a paper to fill out as a lab group, and it'll be relevant to the labs that we're doing in class. We typically hand them out on the results day. So Wednesday for you guys in 4102, Thursday for 1102. And you guys will complete them as a group and turn them in and be graded that way. You also have digital labs that are done through digital labs that are done through McGraw-Hill, which is the connecting. There is a little connect guide on Canvas, so you guys can look through that. And there is actually a first lab report that you guys can walk through on McGraw-Hill that is a digital intro lab report thing. So basically it shows you how to use the digital setup in McGraw-Hill. So you just have to complete that. It's a really easy point. So make sure you guys do that and don't forget. Um, and lab reports, last little bit is lab participation, which is about 1% of your overall grade, includes things like missing lab or coming late to labs. You will lose lots and lots of points for that. So please be aware of that. Also on top of this, if you are doing things like not following safety procedures or not cleaning up things, you can lose points for that. 3 points for protocol violations. So if you're not following instructions or safety issues, -5 points, -2 points for absences, -1 point for tardiness, 20 points in total. So make sure you guys pay attention to that. On top of this, for lab grading policies, if you have a question about why something was graded a certain way, especially quizzes or lab reports, the first thing you want to do is go talk to your TA because they're the one that do all of your lab reports. So if you have a question, you want to talk to them first. Then if you disagree with the reason why they graded a certain way, you can write it on a piece of paper, staple it together, turn it in, and me and the other TAs will read through it and discuss it and decide whether or not you should get points back. However, if you just come to my office and be like, hey, why did it grade it this way? I will tell you, I don't know. Did you talk to your TA? And then you won't get a good answer in terms of question. So make sure you guys do that. Now we're going to talk a little bit about proper lab attire. And this comes in with safety, which we're going to talk a little bit more in just a bit. But proper lab attire is mandatory and will be enforced at all times. This includes things like long pants. So something like jeans or scrub pants, khakis, anything like that. Closed-toed shoes, so nothing like sandals or flip-flops, socks, shirt, and lab coat at all times. You shouldn't wear hats in lab. Hats have anything fall off into things. We don't want to do that, so make sure you guys aren't wearing hats in lab. Goggles and gloves are required at all times. If you're not dressed appropriately for lab, you will not be allowed to work in the lab. I will no problem sending you home to go change, which means that you're going to miss the entire lab and you're going to lose points for that. So please make sure you guys follow that. One really key one that I want to point out for long pants, a lot of people are not cognizant of what long pants are. Make sure that your pants go all the way down to where your shoes are when you are sitting. So if you have a big gap between where your shoe is and your pant leg with a bunch of exposed skin, that creates a hazard for you. If something breaks or shatters on the ground, it's most likely going to break and shatter on the ground. So if it's going to do that, it might shoot out shards of glass or bacteria or anything. And if you have a bunch of exposed skin really close to the ground, it's going to cause an issue. So make sure you guys are paying attention to that and have your pant legs go all the way down. Now, some people do have issues with things like they have like a workout class or a PE class right before lab. One of the things that you can do is you can bring a pair of scrub pants with you and you can put them on outside the lab and then come in and you'll be fine and in perfect lab attire. However, if you put them on inside the lab, that is a no-go because that means that you've violated proper lab attire. So make sure you guys do that outside the lab before you come to lab, okay? Make sure you don't try to like keep anything like scrub pants in your drawer because obviously that would be inside the lab and you have no way to get them. So keep that in mind. Okay. So hopefully you noticed when you go through, there is a little sign out there that says it is a BSL-2 lab. And what that means, it's biosafety level 2. The bacteria are able to cause disease in humans in this lab. That's what that means. Now, generally speaking, they don't cause diseases in humans, but they have the potential to do that. Typically, people at high risk are people that have compromised immune systems. But it's pretty difficult to get sick in this lab. Most of the bacteria we work with are BSL-1 or BSL-0. However, we treat all the bacteria in the lab as though they were BSL-2 at all times. So everything we do in this lab is done under the assumption that it could be dangerous. Make sure that you guys are always following proper safety protocols. We'll go over those more and more each time. Couple of important things. Make sure that if you happen to be a person with increased risk of acquiring infection because you have like compromised immune system or something to that effect, you let me know right away regarding this and we will work out a system just to make sure that you guys are safe. It's really not that big of an issue as long as you're following all the safety procedures that we're doing in class. You're not increased hazard by making sure you're following that. A couple other things to make sure you guys are aware. It is important that students be, if they're like injury or anything like that that you want to let your instructor know right away. The Student Health Center can treat minor physical injuries, so like cuts and burns, things like that, which we are not expecting to have happen. So make sure you guys are aware of that. The health center doesn't provide emergency care. So if something really serious happens, uh, that's going to be a bit of an issue. So you'll have to go to an emergency facility off campus. The university is not responsible for that. So make sure that you guys are aware of that in terms of costs and things like that. But generally speaking, in this lab, it would be very, very difficult to get hurt in this lab. Um, one more thing, each student will be assigned a lab drawer potentially. You guys might be assigned a cubby. If you're assigned a drawer with a key, make sure you guys do not lose that key. The key looks an awful a lot like the university um, on-campus mail keys, the PO box keys. So make sure you guys put it on your keychain right away and label it so that you guys can know what it is. If you guys lose it, it's really hard for us to replace, which will be a $50 fee, and your final lab grade will be dropped by 10%, which is bad, so don't do that. Also on here, we have Title IX information. If you or anybody you know has been discriminated against on basis of sex or gender or assaulted in any way, we do have a lot of resources at the university at the Title IX office. They are very, very helpful, and they've got a great staff there. I've talked with them several times. They're great people, and they have a lot of information. So if you have any need for this, we have the information listed on here so that you guys can contact them. So what does your schedule look like? Schedule is looks something like this. Boy, this view is kind of weird looking. Hold on. Okay. Apparently the normal view thing does not like that. So we're just going to go here to edit view. The way this is set up is a little complicated, I know. For you guys, we don't have class on Monday. So like you're watching this video, that's the reason why. We would normally have this, the second group come in on Wednesday, but since we're all doing stuff on Wednesday, this will be your online orientation stuff. This is the stuff that we're going to be covering today. It's set up by group color. So the blue highlighted parts of this calendar are for the red, blue, and white groups to meet. The gray ones are for the green, yellow, and orange groups to meet. And the orange one is a task that all groups have to do. Now, they don't necessarily all have to do them on the same Friday, but you do have to do them before you come to class. So, for example, you can see that this Friday we have environmental sampling, human sampling, hand washing, and epidemiology lab lecture, as well as the microscopy lab lecture. And you guys can watch through those videos. They will be available for you guys to watch on Canvas. On top of that, the lab safety sheet is going to be due, which I'll talk about in a second. The very first week, which is red, blue, and white group, you guys will meet and you guys will do this procedure. You'll then do the results on Wednesday as well as the microscopy lab. And then at the end of the day, what you guys will have do is the environmental hand sampling, environmental hand washing and epidemiology lab report and microscopy lab report due, and you will turn them in before the end of lab as a lab group. Next, coming up Friday, you guys will have environmental human sampling, hand washing, and epidemiology for the green, yellow, and orange group. So you guys will have the same kind of lab lecture. But a key thing to note is that you guys will be having your first quiz, which is quiz 1 on this day. So it's really, really, really important that you make sure that you guys complete quiz 1 online on Canvas. So this will be for everybody. So everybody's doing quiz 1 on this Friday. Then you guys will do environmental human sampling in the green, yellow, and orange groups that second week. So this is the A week, this is the B week. B week starts this lab, and they're going to basically do the same thing, and your guys' lab reports will be due on the second part. On the 5th, February 5th, you can see that we are going to then do culture transfer techniques and smear prep. You will have quiz 2 microscopy and quiz 3 environmental human sampling, hand washing, etc. So you guys will have those 2 quizzes due as well. And so it'll basically go on like this for the entire time that we're doing this. I know it's a little confusing. If you have a question, make sure to ask me or your TAs. That's just kind of the way it is, unfortunately. Okay. So next little bit. Safety stuff. There is a safety rules page in your lab manual. It's page 2. It's got a whole bunch of stuff on it. And so we're going to talk about that really quickly. And there will be a digital version of this you will need to sign and turn in on Canvas. And it is worth a lab grade. You will need to submit it to your TAs, and then they will be able to um, verify that you have read through this before you guys can come back to lab. So make sure you guys finish this before you guys do it. Really quickly on page 2, I'm going to cover a couple things. The first one is never place books or backpacks or purses or personal items on the bench tops. Oops, skipped ahead there accidentally. Everything should be put on these pull-out drawers. The drawers are on the side of where you sit. So they look like a little tiny drawer to your left or to your right, depending on where you sit, and you pull it out and there's actually a little table there. That's where things like pens, pencils, etc. like that go. Book bags and purses and jackets and anything like that, you never ever want to have on the floor or on the chairs. That creates a tripping hazard. You guys want to put it on the cubbies, which are right in the door when you come in the lab on the left-hand side. So make sure you guys put them in there when you come in and then you sit down. The next thing, never ever bring any electronic devices in the lab, which typically is just cell phones. A bunch of these other things, MP3 players, radios, what have you, not typical things that we have anymore because they're all replaced by cell phones. So just make sure you guys leave your cell phones off so it's not ringing in your cubby and not in your pocket. Don't take it out. If you take it out, we have to decontaminate it because it's potentially contaminated. I give you the option. We can either flood it with bleach for 30 seconds, which it does not survive, or we can put it in the autoclave at 121 degrees Celsius for 15 to 20 minutes, which again doesn't survive. So don't do that. That's a bad idea. Make sure you guys keep your cell phones away. Clean your work area with dilute bleach solution at the beginning and end of each lap. Now, it says in here dilute bleach. We've actually switched it over to something called Cavicide, which is a alcohol-based spray with other surfactants. It is not just it's not just alcohol. So it's got lots of things in there to kill bacteria and a variety of viruses as well. It is very, very effective. You want to spray it down. You know, you don't need to go crazy with it. Just create a nice even area. You'll take paper towels, wipe off the table area that you're sitting at, and throw the paper away in the trash cans, which are found at the end of each lab table. Now, really quickly here, and this is actually very important. You want to do this at the beginning of lab because you just don't want to trust the person who was here before you. Maybe they were a dummy and they left a big contaminated mess, so you always clean it at the beginning of lab. You also always do it at the end of lab. It's the last thing you're going to do before you leave lab because you don't want to be a big dummy that left a contaminated mess for the next people. So make sure you guys are always cleaning off those tables whenever you come in and at the end of lab. Okay, important safety stuff aside from that. You should always wash your hands with antimicrobial soap when entering and leaving the lab. So you do this at the beginning of lab just so you're not bringing any bacteria from the outside environment to your working area. And of course, you want to do this at the very end of lab because there are bacteria from this lab that we don't want going out in the outside environment. So make sure you guys always wash your hands at the beginning and end of each lab. And there's lots of soap and throughout the entire lab. You can wash your hands at any sink except for one, which you guys will see in the lab tour video in just a second. Wear a lab coat at all times, preventing kind of accidental spills or things like that. Make sure you're not wearing anything like shorts or capris, dresses, skirts, sandals, etc. in the lab. Long pants and shoes that cover your entire foot are required. No hats in lab. Make sure you guys leave your hats in the cubbies. If you have long hair, it's a really good idea to tie it back. A lot of people will keep a hair tie in their lab drawer. Obviously, you wouldn't want to be looking over at something you're working on and have your hair fall into bacteria, which is gross. So don't do that. Gloves should be worn at all times during staining procedures and handling bacteria. Lab gloves, when they're disposed of, will go in the biohazard bag, which I'll show you guys in the lab tour video. Do not place anything in your mouth or eyes while in lab. This includes food, no eating and drinking in the lab. If you have anything that's like water or a snack or anything like that, you guys can actually just leave it outside the lab door. We do that all the time. You'll see my coffee cup out there all the time. Nobody messes with it. It's just something you get used to when you work in a lab like this. On top of that, you also don't want to have anything like gum or cough drops. People tend to forget about those when they're coming to the lab. Make sure you guys spit those out before you come into the lab. Also, do not apply cosmetics in lab, which I know sounds really weird, but I've actually seen somebody trying to put on mascara first thing in the morning in the lab, in the back of the lab room. Please don't do that. That's a terrible idea. Other things that people tend to forget about are Blistex and chapstick. Don't put that down in lab. Never pipette anything by mouth. We have mechanical pipettes. We'll show you guys actually how to use those later on in the semester, but never pipette anything by mouth. Contact lenses shouldn't be worn in the lab, but I'm not going to go around and like check your eyes. What you should do is be aware that contact lenses carry inherently higher risk in this lab. I wear contacts all the time, it's totally fine, but you need to be very careful not to touch your eyes in lab, especially contacts. Some bacteria that we work with in this lab can actually grow in the matrix of contact lenses. They can cause a severe infection and can even cause you to go blind. So you never ever want to touch your eyes in this lab. If your contact happens to roll under your eye or something like that, while you're in the lab, what you want to do is get up, wash your hands, leave the lab, fix it, and then come back and wash your hands again. Never ever touch your eye in lab. So that's just an increased hazard that you should be aware of by wearing contacts. Number 7, do not remove any media or equipment or bacterial cultures from the laboratory. I know it sounds crazy, but people have asked to take a plate home to go culture their sink at home, in the kitchen. No, don't do that. We're not going to do that. Anytime you carry culture test tubes, make sure you guys use a test tube rack. There are actually racks at your table. These should be returned back to your table every time that you guys are done with them, except for the one that goes in the incubator that's assigned to your lab group. But anytime you walk around in the lab, you always want to carry test tubes in racks because test tubes are notoriously round and notoriously slick. And because there's not a lot of room in the lab, you can easily bump into people and drop them. And of course, you wouldn't want to drop bacteria on somebody else. So make sure you guys always use a rack. Okay, number 10, immediately cover spilled cultures, broken test tubes, paper towels, and then saturate them with the disinfectant solution. We're going to change that a little bit. What you're going to do is you're going to first let me or your TA know right away, and then we will go and fix it together. The worst thing you could ever do in this lab is not spill or break something. This is a teaching lab. Sometimes spills happen. It's okay. We're not going to be mad at you. But the worst thing you could do is have that happen and try to fix it yourself and do it wrong. And now we have an unknown contaminated mess going class after class after class for weeks maybe because we didn't know that there was a spill somewhere. So make sure you guys let us know. We won't be mad at you. We'll just work together to clean it up. It's totally fine. On top of this, make sure you guys report accidental cuts or burns to the instructor immediately. Let us know if any of that happens. At the end of each lab, make sure you guys put culture materials in the proper disposal area, and we will cover that. Now, there's some other OSHA information which I will talk about in the lab tour video, so wait for that. But at the very end of this, you guys can see a signature page and a date. I'm actually going to give you guys a digital version of this, which you guys will digitally sign. You guys can use Adobe Reader, I believe, to sign these things, and then you guys can turn them in. You'll just email them, just turn them into Canvas, and then you guys will be done. Okay, now, what we're going to do now is going to show you guys the next part, which is the reason why we're covering all this safety stuff, and then I'll show you guys a lab tour video. There is a on Canvas a thing called CDC Salmonella case study, which is actually a very interesting case study. If you don't know what case study it is, usually when a bunch of people get an infection, the CDC will release a report kind of covering it so that we can learn from these kinds of issues. Now, typically when we have salmonella, we're thinking about things like infected chicken or food, some sort of infection source like that, where a bunch of people get sick from like a local area. Here we have an interesting one because this one doesn't really follow that. So if you look at the infection map here, usually when you have a bunch of people infected with this, they might be in a small area like a city or a local county or a tri-state area that's like a local chicken supplier. But this one spread across the entire country, which was very unusual. And so what they ended up doing is they went and looked through it and they found that there was actually one common source for all of these, and it was at a teaching lab. The way they were able to figure that out is they do this thing called pulse field gel electrophoresis, which is a type of genetic fingerprinting where we can get strain specific information on salmonella. So when you go into salmonella with the salmonella illness, your doctor, the hospital will go, oh, hey, that's a reportable disease. So take a sample, report it, and send it off to be pulse filled gel electrophoresized, tested to see what kind of strain it is so that they can maybe tie down multiple cases. Because this is all reported to the CDC, that crosses state lines, that allows us to get all this information and kind of figure out stuff that might not be easy to connect. In this case, these were all connected to a strain of salmonella that is used exclusively for research purposes, but is also sometimes used in teaching labs. And what they basically found out is that one teaching lab was not following proper safety protocols. They spread across the country. People went home from classes and spread it all over. One of the things that you can see that typically shows up, by the way, this is what the pulse filled gel electrophoresis looks like. You can see these slight variations in the patterns here. These slight variations kind of show small differences between these different strains. So you can see these little strains. It's actually a very, very handy technique. But you can see that these are actually all related. These are all the same genus and species, but they're variations within the strain. So they all have small, uh, tiny little variations within them. So you can see how all of these ones, so these, I think it's 10 organisms, are all identical strain-wise, as are these ones. But then these ones, you guys can see tiny little differences. So for example, this guy isn't exactly related to this guy because it's got an extra band right here. That means it's got a slight variation in its genome. So it's a very, very useful technique. But when we look at the number of people infected, normally when we see an infection that's an epidemic, so a sudden spike in the number of cases, it'll slowly taper off as that contamination source goes away. But in this case, it kind of had a low-hanging contamination rate through a very long period of time. And that was actually very unusual. And that's because the class kept having these issues throughout the entire teaching time. So a lot of people would get this, it just wasn't getting out of the system because they were constantly reintroducing this contamination issue. So what did we find out? Well, we found out that the median age was about 21 years old, so college age, which is what we expect. Most of you guys are college age, so that makes sense. However, the age range was very extreme. So it was less than 1 year old to 91 years old. And generally speaking, when a normal, healthy adult gets salmonella, they just feel really bad. They might not even realize that they have salmonella. They won't go to the hospital or anything like that. They'll just feel really bad for a day or two, and then they'll be fine. However, if you are very, very young or very, very old, you have a couple of overlapping issues, which is weakened immune system or inexperienced immune system. You might not have enough body fat or other health conditions that contribute to this. If you're a baby, you might have an inability to absorb enough nutrients if your intestinal lining is shedding because of the salmonella. Lots and lots of problems associated with that. So for those people, it's actually incredibly dangerous. Very, very, very serious issue. And you can see that even one death was reported. Generally, it doesn't kill people, but it can. And it's probably one of those two people that are on the extreme end. About 12% of patients were hospitalized, so you can have very severe reactions to this. 12% is a pretty high number out of 109 individuals, so there's a pretty hefty amount of people got sick from this. What do we take away from this? Well, what they found out was the lab was not following proper safety procedures. Uh, they weren't doing basic things, and it's very crucial that we enforce rules for this reason, because we do not want to do this. Most of you guys are already in some sort of health industry or trajectory for your career. Many of you are going into medicine, PA, doctor, dentistry, all sorts of things. And I know that for a lot of you, especially those of you who also maybe have a job on the side where you are working in a hospital or in a doctor's office or in a nursing home, the worst thing for you guys in the world would be to be the reason somebody else got sick or maybe even died. So we want to be very careful in this lab not to take anything out of this lab. That's the reason why we harp on safety rules all the time. Washing your hands before and after lab, decontaminating your work surfaces, making sure that all of your book bags and things like that are actually in the bookshelves, making sure that you guys dispose of media properly, making sure that we one that a lot of people don't think about is actually talking about safety. So we're doing safety training right now. We're actually going to do some more safety training, which I'll talk about in a second. And then we're also going to be doing things like safety. Every single lab lecture we have, we talk about safety and potential hazards and what you should do for anything like that. Other important things is making sure that we're enforcing the rules. So rules are great right up until nobody is enforcing them. So we enforce them very heavily, especially during the first week, so you guys can get used to doing the rules, and so that you're getting to an automated state. You're not thinking about having to do them or forgetting them. Rather, you automatically do them without even thinking because you already know them so well. Other things are making sure we're not using excessively dangerous bacteria. So we don't use salmonella lab. We can get the same results with other bacteria. It's just an increased hazard that we don't need, so we don't use it in our lab. But it's very important to make sure you guys go through this. Now, we also have a safety training through Baylor. It's on this website called BioRaft. Me and your TAs generally do this as well. We go through all this stuff. It's actually very, very intensive training that covers a broad variety of things, but we also have a basic version. And that basic version is the one that we will actually give you guys. There's a PowerPoint that's listed on Canvas that you guys can download and use. That's the basic biosafety training. You can download that, walk through the PowerPoint slides, and then you guys will take the basic biosafety quiz. Basic biosafety quiz is also going to be your first run at doing the webcam stuff, the lockdown browser, which you guys are probably all used to by now, but just make sure that we get all kinds of things worked out with that. What I want you guys to do is make sure you take that basic biosafety quiz before you guys come to lab on the very next week. So when we do epidemiology lab, make sure you guys have already had that basic biosafety training completed. This is the same training. I literally stole it from the Department of Environmental Health and Safety at Baylor. It's the same basic biosafety training you take in BioRack. So make sure you guys run through that and complete that. Aside from all that, what we'll do right now is we will do our lab tour and talk about a couple other things like the OSHA requirements for things that you need to know in lab, and then you guys will be all done. So 1 second.
All right, here is the lab. A couple of important features. We have lots of sinks for hand washing in the back. You can see that every sink has soap on it so you can wash your hands. That's an important part of the lab. You want to wash your hands before and after lab. We also have a special sink over here. This is the eye wash station sink. This sink is special because it's the only sink for one purpose, and that's for washing eyeballs. So that would be this guy over here. To use it, you just actually pull this over, flip it down, and push the paddle here, and hold this up to your eyeballs, which feels unpleasant, and you'll do it for about 10 minutes or so, 5 to 10 minutes, and it'll flush anything out of your eyes. Now, of course, we won't need to use this as long as you guys are following basic safety procedures by having your goggles on. But most important thing, this sink, only for washing eyeballs and nothing else. All the way over here, we have a couple other items. OSHA requires you to know these, which is where the MSDS sheets, or SDS sheets as they're currently known, are. We also have our first aid kit, which is this guy right here. First aid kit also has lots of band-aids in it. If you happen to come to class and you maybe have an open wound and you don't want to do a micro class with that, just let us know. You guys can get a band-aid and some antibiotic ointment out of there and keep it covered up. Lastly, in this corner, we have the spill kit. Spill kit is very important. It is something that we have to have in the lab, even though we won't really work with chemicals in high enough concentrations to require using it. You do have to know where all these items are for OSHA. If they come in and inspect us, they can ask you, and you have to be able to know where these things are. MSDS sheets, by the way, are sheets that have all the data for every chemical in the lab. You guys don't really need to know this, but we need to have this, know this. But if they ask you guys need to know where it is. All over here by the instructor's desk is the glass disposal box. This box is for disposing of glass, but don't throw anything in here unless you ask us first. So if you break something that has bacteria in it, we want to make sure to decontaminate it before we put it in this box, so make sure you let us know, and we'll handle it together. All the way over here we have our chemical safety shower, which is this guy. Now, this one is really interesting because when you pull the handle down, tons and tons of water will come off, and you'll take off any area of affected clothing that might have chemicals on it. So if you spilled chemicals on you, then you'd pull this and do this. Well, you won't really have to do this in this lab because we don't deal with volumes great enough or things dangerous enough to warrant the use of this. And you definitely don't want to pull this handle unless we're absolutely sure, unless we're telling you as TAs or unless I am, because there are no drains in the floor. So it'll actually destroy all the floors and the lab below us, and we'll be really sad, and it'll be shut down for a couple weeks while they fix it, which is not good. So make sure you guys don't do that. Also over here we have the fire extinguisher, which we have training for the TAs as well as myself. You guys don't need to know how to use it, but you guys do need to know where it is. And we also probably shouldn't be having any fires in lab. We also have this guy right over here, which is the biohazard bin. This is where you dispose of things like gloves and plates. We tape them up in stacks and put them in here. You'll be using this a lot throughout the semester. We also have over here our autoclave cart. Autoclave cart is for disposing of tubes. And you guys can actually see we got two different types of tubes on here. We've got one's in orange rack and one in blue rack. Blue rack is for broths. Orange is for augers. Make sure you guys keep those separate. That's very, very important to do so. We will remind you guys when it's cleanup time, we have to autoclave them separately. But those are important things to make sure you guys remember. Now, lastly, just to make sure that you guys were paying attention to this whole video, especially the safety training part, we have our special lab mascot, which is Biohazmat Yoshi, which is right over here. And when you guys come in for the very first day of lab, because you of course won't be here first week, you will come over here I'll have a sheet with names on it, and you just write your name on it before you guys leave. And you guys will get extra credit points on your very first quiz. So make sure you guys are paying attention and following instructions. And also don't share with anybody who wasn't paying attention. All right, guys. I'll see you guys in class. We'll have more discussion about things like drawers and all that other stuff and all the details for labs. So I will see you guys then. Let's have a great semester.